Welcome back. I'm Cameron Rowe. In today's episode of Biology First, we will be talking about a disease many of us may not know, Wilson's disease. We will discuss the story of a woman who through her struggle has become a beacon of hope and whose determination to spread awareness about the disease has inspired many. The focus of today's story is a woman named Gina Genotype, who is ailed by Wilson's disease and her pathway from diagnosis to overcoming the illness. First, we invite Dr. Diaz to help us get a general overview of what Wilson's disease is and how to recognize its symptoms.
This disease is a rare genetic disorder characterized by excess copper stored in the various body tissues, particularly the liver, the brain, and the corneas of the eyes. Some symptoms of this disease are scarring of the liver failure, neurological problems, and kidney problems. Alongside these are the psychological problems like personality changes, depression, irritability, bipolar disorder, and psychosis. The PRMP gene provides instructions for making the prion protein, which is active in the brain and other tissues and takes part in the transporting of copper. When this protein stops functioning, there will be a buildup of copper.
When Ginta Genotype began dealing with some troubling health issues a few years ago, Wilson's disease wasn't something that ever occurred to her.
The first symptoms I started to have was balance issues, and I'd fall down a lot and trip over anything. That's when I started to know something was very off.
Her health got progressively worse.
I started to get really tired. I'd lay in bed like half the day. I was starting to get weak too, and then my swallowing went out. It kind of feels like you're stuck in a body that's not you. It's a scary feeling to have.
During a routine eye exam, she finally learned what was going on. Her optometrist suggested she might be dealing with Wilson's disease.
He said he noticed like color differences in my eyes, and he saw like stuff floating around my eye. That was the copper. It's scary to hear that.
Now we are going to talk to Gina's optometrist, Dr. Diaz.
I am Dr. Diaz, and I am Gina Genotype's optometrist, and was the first to recognize her symptoms of Wilson's disease. She came in for a routine checkup, and I immediately noticed the yellow tinge to her eyes and the copper deposits. I told her that I had recognized something called a Kayser-Fleischer ring, or a KF ring, which is basically a large ring of copper deposits around the iris of the eye. I told her that she needed to seek immediate medical help from Wilson's disease specialist, because as far as I could tell, she definitely had the illness.
In order to better understand what's going on inside Gina's body, we need to first take a look at how proteins are made. We are going to visit with Dr. Cadiella, a Wilson's disease specialist.
The first step to make proteins is called transcription. Transcription is when RNA is made from DNA. The information is copied from one molecule to the other. The DNA sequence is copied by a special enzyme called RNA polymerase to make a matching RNA strand. The product is called messenger RNA or mRNA because it carries the genetic message from the DNA to the protein-making machinery of the cell or the ribosome. The mRNA codes for amino acids to make proteins. There are three main stages for translation, which is the second step in the process of making proteins. The first stage is initiation. This is where ribosomes attach to the mRNA molecule of the start codon. The ribosome can enclose two codons at a time. A codon is a group of three base pairs which code for an amino acid. The second part to initiation is that each tRNA carries three base pairs called an anticodon, which bond to their complementary codon on the mRNA. The second step is elongation, which is the addition of amino acids to the growing polypeptide chain. The tRNA keeps attaching to the adjacent mRNA codon, as a result adding more amino acids to the chain. The bond holding the tRNA and the amino acids together is broken, and a peptide bond is formed between the adjacent amino acids. As the ribosome can only cover two codons at a time, it must now shuffle down to cover a new codon. This releases the first tRNA, which is now free to collect another amino acid. The third and final step is termination. The polypeptide chain elongates until the ribosome attaches to one of the three possible stop codons: UAG, UAA, or UGA. The gene that gets mutated in Wilson's disease is called the ATP7B gene, and the ATP7B gene is located on chromosome 13. The type of mutation is a nonsense mutation, And the DNA changes from CAG to TAG, which causes the amino acid glutamine to change to a stop codon, which shortens the protein. So here it's a picture of the ATPase2 protein, and this is how it looks like when there's a nonsense mutation of the ATP7B gene, so the protein is essentially incomplete and it does not function properly. So without the protein working, you can get symptoms associated with Wilson's disease. The ATPase2's main function is to transport copper to the Golgi apparatus, which then either uses the copper safely or disposes it. So when there's a mutation in ATPase2, it cannot do its job, and thus there's a copper buildup that occurs. Also, like a quick fun fact is that other mutations can also happen at the ATP7B gene, like a deletion or insertion mutation, but these mutations won't give you Wilson's disease. Instead, they give you a milder version of the symptoms. The Wilson's disease primarily affects the liver and brain cells due to the accumulation of excess copper caused by the mutation of the ATP7B gene. The ATP7B gene provides instructions for making a protein called the copper-transporting ATPase2. Copper-transporting ATPase2 is found primarily in the liver with smaller amounts in the kidneys and the brain. It plays a role in the transport of copper from the liver to the other parts of the body. Copper-transporting ATPase2 is also important for the removal of excess copper from the body. Within liver cells, copper-transporting ATPase2 is found in a structure called the Golgi apparatus. Here, the copper-transporting ATPase2 supplies copper to a protein called ceruloplasmin, which transports copper to other parts of the body via the blood. The second function of ATPase2 is to package excess copper into vesicles to excrete via the bile. The Wilson's disease causes a mutation in the ATP7B gene. The defective gene leads to markedly decreased functional copper-transporting ATPase2, which restricts copper from entering the Golgi apparatus. As a result, the copper isn't allowed to bind with the ceruloplasmin from where it would have been safely used by the body. Instead, the copper becomes free copper. This free copper reacts with the hydrogen peroxide in the body to produce a hydroxide radical, which is responsible for cell, tissue, and organ damage.
Wilson's disease is an autosomal recessive disease, which means that in order for someone to inherit the disease, both parents need to have the recessive allele. As you can see, this is the dad's chromosome and this is the mom's chromosome. In order for the child to inherit the disease, the child must get the recessive Wilson's disease allele from both parents, which is represented by these two little A's.
And now these are some possible crosses.
The first possible cross is a Punnett square that represents a heterozygous male carrier and a female heterozygous carrier. And you can see that their offspring, that 75% would be unaffected, 50% would be carriers, and 25% would actually have Wilson's disease.
This Punnett square shows that the two parents have Wilson's disease and what the likelihood of their offspring getting Wilson's disease is.
And this last Punnett square shows a dad who has Wilson's disease, who mates with a mom who's a heterozygous carrier for Wilson's disease, and we see that 75% of the kids, there's a 75% chance that the kids are unaffected, 50% chance that they'll be carriers, and 50% chance that they'll actually have Wilson's disease.
This is a Wilson's disease pedigree. It shows how the recessive trait of Wilson's disease can be passed through a generation. There are 6 symbols. The unshaded circle shows a non-carrier female. The unshaded square shows a non-carrier male. The striped circle shows a carrier female. The striped square shows a carrier male. The shaded circle shows a diseased female, and the shaded square shows a diseased male.
From the beginning of the pedigree, you see the example of a heterozygous carrier female mating with a male who is not at all a carrier for the disease. As you flow through the generations, you see their offspring, which are 7 females, 2 of which are heterozygous carriers. And as you flow through the chart down, you see certain carriers, 1 male and 3 males, until you get to the second and last generation, which has a heterozygous carrier male. And then finally, you see a first case of someone affected by Wilson's disease until you get to the last generation, which has a male and a female offspring who are both affected by Wilson's disease.
And the reason why this proves that Wilson's disease is indeed a recessive trait is because if a trait is dominant, then you can't be a carrier for it. And since Wilson's disease is a recessive trait, there are carriers for the disease. And it makes sense because if someone is, so if the trait is dominant, the reason why it can't be carriers is because only one dominant trait is required for it to mask the recessive trait. So. Hi, I'm Dr. Kadiala and I'm a Wilson's disease specialist. So, as a doctor, I recommend taking medications called Chelting agents, which bind copper and then they prompt your organs to release the copper into your bloodstream. The copper is then filtered by your kidneys and then released into your urine. Medications like penicillamine and trientine serve these sort of purposes. Another alternative is zinc acetate, and this medication prevents your body from absorbing the copper that you eat through food, and it's typically used to prevent copper from building up again after treatment with penicillamine and trientine. And for severe liver damage, we recommend a liver transplant. And if you do have Wilson's disease, it's essential to avoid foods with high amounts of copper, and some of these foods include liver, shellfish, mushrooms, nuts, and chocolate.
We thank you for watching today's episode and following us through the heartfelt story on Biology First. Next episode, we will be discussing more genetic diseases and their effect on the life of the people that have them. Thank you for watching, and we will see you next week on the following episode of Biology First.